Everyone. So I get to introduce Helen tonight. Helen grew up in Massachusetts. We're lucky enough that she decided to come here. But while she was growing up there, she fell in love with gardening. And when she was in school, she told her professor that she wanted to be a landscape architect. And they said that that was not a job for a woman. And she's just blown everything out of the water. So I don't think that they were right. She just retired last year after 31 years at BGSU, where she taught botany and restoration ecology courses. She's also been a member of Wild Ones for years. In 2020, she helped to implement a sacred grounds garden, and she also participates on our programs committee. Where in one of our meetings, she allowed me to sign her up to give a talk. So I will hand it over to Helen.
Thank you, Sarah. Let's see. So I've kind of been in technology hell all day, trying to get bits of this talk to work out right. So if there are little delays between slides, it's not you, it's not the full moon, it's not the election, it's just my computer. Okay. Oh, so technology hell starts already. It won't advance now. It's actually on, but for some reason this was working earlier and it is not now working. This is very disturbing. I'm totally and utterly mystified. I have no idea why I cannot advance my slides. It just occurred to me that it probably wants me to do this. Yes, how annoying. So much for having my remote. Oh, well, what do you know? Now it'll work. Okay. All right. Let me get this out of the way so I don't have my back to you the whole time. There we are. So title is self-explanatory. Local genotype is something that we hear a lot. And I see a lot of confusion about it. And so I thought this would be a worthwhile talk. I hope that it will inform. I can't promise you will leave here less confused because there are still many unknown issues in how to think about local genotypes and how to use them in native plant restoration. So this is one of the documents that you can find on our website. Everybody write that down, by the way, but it's easy to find on the chapter website. And for those of you who are at the back and can't read this, I'll just jump to the chase here. Native plant species is one that occurs naturally in a particular region, ecosystem and habitat, and was present prior to European settlement. Local genotypes are native plants and seeds that originate from local or regional sources with similar environmental conditions. I want to emphasize that because we're going to return to that. It's really important. Wild Ones advocates selecting local genotype plants when possible to grow in your planted landscape to sustain biodiversity and ecosystem health. So where did this idea come from? People who study native plants recognize the fact that some of them have really wide geographic distributions, like our budding big goose, one of the predominant grass species that we've got in our prairie ecosystems. And I was really surprised when I saw this. I had no idea that it reached all the way to this species. Given a species with such broad geographic distribution, it's easy to imagine that environmental differences between populations in these different places are going to cause evolution of distinct ecotypes that are adapted to specific climate, light, soil organisms, whether it's bugs or microbes, or soil chemistry, herbivores, or other living conditions. So ecotypes are a big part of understanding this. The presence of these specific adaptations is sometimes called local adaptation. And inherent in these ideas is the assumption that seed sources nearest a planting or a restoration site are therefore more likely to be better adapted to that local environment. This is one of the take-home nuggets, okay? And that adaptations of ecotypes, the things that make them adapt and that have a genetic basis. Okay, so if that's what you wanted to know, what are local genotypes, you can leave it. Okay, this is take-home message from me. Oh, here we go with the slow animation. So a local genotype refers to native seeds or plants near a planting site that have a genetic background that is typical for a region and that are adapted to that region. So anything that is considered local genotype, those are going to have some shared genetic background and shared evolutionary history. And therefore, the local ecotype refers to plant sources with similar climate and ecology to this site. Slight differences in the uses of the words. And they're the local ecotype because they've had this long period of adaptation to the local environment, and therefore they are a good place to put the bets. Okay, they're more likely to be able to do well and not just establish, survive, and grow and reproduce, but also provide ecosystem services. That is, to service, for example, pollinators, okay, or provide other ecosystem services that make ecosystems functioning as whole entities. So, where's the evidence for the presence of these locally adapted genotypes? Is there a scientific basis for this idea? Well, it's actually fairly old. This was one of the earliest studies done in ecology by Pleasant-Peck and Hesse. And they did an insane amount of work. They worked in California along this elevation gradient from sea level up to something like 3,000 meters in elevation, and then of course it goes down the backside of the Sierra Nevadas. And what they did working with yarrow, only not our activated yarrow, but a mountain variety of it, variety lanulosa. And they worked in establishing 3 common gardens in these places here, where the blue stars are. And in each of these 3 common gardens, they grew seed that had been collected from all of the individual populations at these different elevations, different geographic localities. And if you look carefully, they're morphologically really different. Some of them are short, some of them are tall. They clearly have different phenotypes, different appearances. And it's reasonable to assume that that has something to do with where they're from. But the real question is, what's the evidence for that? So they found that when they took seed from each of the sites and planted them into each of the 3 common gardens, that plants that were from high elevations were always shorter, right? So they maintained this shorter stature. But they also grew better than low elevation plants in the high elevation plants. And the same, because this is a reciprocal kind of experiment, the same result, if you will, the same parallel conclusion applies to them in the low elevation plants. So these things can happen because there has been a genetic change in a population in response to geographic change and difference in selection pressures because of the ecology of where these organisms live. As a consequence, these are all considered to be locally adapted ecotypes. Notice they're all considered part of one species, and they're considered part of one variety, the variety lanulosa within one species. So these are not different entities taxonomically. They are interbreeding. They're capable of setting offspring when they are hybrids. So locally adapted genotypes, that's how the idea was first developed. Okay, so the second question is, well, what exactly is local? How do we measure it? And is it always the same kind of local? Turns out that a lot of the time people like to use a distance guide. They will say, for example, don't move plants more than 20 or 50 or say 100 miles from their original location. And you can see there's quite a variability here in what people use. So miles rules like this tend to be inconsistent and frankly a little bit arbitrary. Turns out there's no scientific basis for any one of these numbers that you can uniformly apply in all situations and across all species. So this is confusing, not just to people who are just starting to learn how to deal with native plants, but even to the scientists. So as a consequence, distance and defining local according to the distance is just not the best practice. And as I'll get on and tell you in a bit here, there's other things that we should be thinking about as well. But one of the best summary statements is here. For plants, local is best defined ecologically in terms of climate and environment rather than miles. So if climate and environmental or ecological conditions are fairly uniform across 100 miles, well then maybe that defines the local. But that may not be the same for all species, and it may not be the same in all parts of the country, hence the inconsistency and the confusion. So what I wanted to do was to share with you some nuances, some reasons why this is not straightforward and why local, as defined by, say, nearby, is not always best. So this work is about 20 years old, Montalvo and Elstrand, again, working in California. They were working with California deerweed, shown here. It's a legume, changed names a couple of times, and we don't really care too much about the specific name there. But they collected seed from 11 different populations of this legume shrub. And they had the help, the money, and energy to do genetic studies on each of these 11 populations. And that's a lot of work. It's very expensive as well. And then they grew seedlings from each of these 11 populations in 2 different common gardens that were ecologically different. And then they measured the fitness, how well they grew and how much reproduction they did after 1 year. And in this case, they found that these measures of how fit or how vigorous the plants were was correlated with the genetic similarities between the populations and the similarities of the environments of the populations, but not geographic. So it kind of reinforces this idea that, you know, the plants are listening to the ecology and don't really care how far away from us. One more. So here's the take home for you, right? These are scientists running around in California doing fancy science. What does it mean for you? Well, the most important thing I would say is know something about the ecological conditions of your site, whether it's a chunk of your backyard or a big swath of 6 acres that you want to turn into prairie. You need to understand the ecological conditions where you're trying to establish these plantings and to use that in part not only in deciding what plants to put in there, but maybe where your seed sources should come from. Just because you can get seed of some of these plants from down the road doesn't necessarily mean that those sites are the best places to collect seed from. It's the ecological similarity that you should be thinking about. Does that make sense? So, because of some of these ideas, in more current thinking, people have developed on large scale thinking guidelines for where it's okay to move seeds around and obtain seeds for plantings that are based on two factors. First, something called seed transfer zones. Seed transfer zones are geographic areas within which seeds are thought to be able to be moved around with minimal risk of maladaptation. They do okay. They're adapted as long as you are working within these seed transfer zones. And so, what are these seed transfer zones and how do we make use of them? First, you use what are called provisional seed zones. So people gathered a whole lot of climatic data. And in particular, when they figured out what climatic variables would be most informative, they decided that winter minimum temperature and aridity in a location were going to be the most useful. So this sounds a little bit like, USDA's hardiness zones, but it's more than that because it also has something about moisture availability in there, not just precipitation, but does it all evaporate again? It doesn't do much good getting a fair bit of rainfall, but it doesn't hang around because it evaporates all the time. It goes up. So combinations of winter minimum temperature and aridity were used to create this beautiful map. Everybody read this one, can you figure out really hard to see. But for moving forward, let's just say that wherever you are, you can kind of figure out what the provisional seed zones are in terms of these minimum temperatures. And then you overlay upon that another piece of data that has been developed by people that I'll tell you about in just a second, something called ecoregions that are specified on two levels of detail, if you will, what are called level 3 or level 4 ecoregions, and I'll show you these in just a second, to refine more specifically within a provisional seed zone where you can move seed. So here is ecoregions of Ohio. So you can't really see ecoregions at the top because I clipped a little piece from Michigan and kind of talked again about where it would be joining us, because being oak openings chapter of Wild Ones, of course, we're all really interested in occurrences of oak openings type vegetation that might happen to be just over the border. And so you can see those over there. But the point is that ecoregions have ecological, not political boundaries, and these have been delineated by the people far wiser than me, EPA and USGS. Lots and lots of people have worked in developing these. And as a consequence of the combination of using this provisional seed zones and the ecoregions, it's reasonable to suggest now that you can use this information to think about where you can move seeds and where you can obtain seeds from. And as a consequence, it's better to use a seed source from your ecoregion, but outside your state, than a source from a different ecoregion inside your state. And so up at the top, we have this little blob of Michigan that has this dark green. Let me back up a second. But the heavy lines on the map correspond to the level 3 ecoregion delineation. And then the slightly finer lines that have little different color blobs within the bigger heavy line delineated regions. Those are the level 4 more detailed regions. So the oak openings actually is part of the ecoregion labeled 57, but 57 is subdivided into 57A, B, C, and D, as it turns out. The oak openings itself, those of you who are on Zoom at home, I'd have to use my cursor to show this, but if you're in the room, okay, this here and this here, these are part of, there was originally openings vegetation there, but this is the current corresponding area of the openings. So they can get it there, but here are other pockets of oak savanna vegetation across the board condition. So those are level 4 ecoregion definitions. But based on that, it's certainly the case that this entire area, okay, the level 3 ecoregion would be a better place to get seed from than anything else in the rest of the state. Okay, so that's kind of an extreme example, but that's how these things work. Not only here, but everywhere. So the more closely we match the environmental conditions with the source of plant material in the planting cycle, the better they will grow. And maps like this help you make those decisions and choices. So if you move away from here and you go to another part of the country, you can use the same approach to help you choose how to do native plants in where you go. So these are kind of large scale guidelines as to how to define what local might be, at least on first pass. I want to share with you some research on one of my very favorite plants, partridge pea. Many of you are familiar with this lovely annual in the bean family. And it was studied by some people that collected plants from populations of this species in 3 different regions: Maryland, Kansas, and Illinois. And in each of these 3 regions, they collected from populations that were at known increasing distances apart. So the distances ranged from 100 up to 2,000 meters away. And as a consequence of this, they then would have, a local population plus a series of not quite as local, nearby other populations along a distance gradient. And so this study structure was replicated in 3 different parts of the country. So again, this is another species like big blue stem that can be found across a pretty broad geographic region. So within each of those 3 regions, they grew seed, both the parents and the offspring of those parents, the next generation, in what was called a home field population. So the collected seed from a site, planted the seed back in that site, along with the seed from the populations that were, say, 100 meters away, 200 meters, 500, 1,000, and 2,000. All of that was all in one place, this home field, in sites like that in Maryland, Kansas, and Illinois. And they repeated this for 3 years. This is a long experiment as far as science is concerned. It takes a long time to do this. In this case, they found what's called a home site advantage. That is, the seeds that came from the place where the plants were actually grown did better than anybody else. Only you got out past, say, 1,000 meters. Now, first of all, 1,000 meters is a little more than a half a mile. Okay, so closer than a half a mile didn't seem to matter. Everybody did about the same. But as little as a half a mile away, the plants that were in the home field were doing better than the ones that came from a little more than a half a mile away. That's extraordinary. Like, the phrase cats see things we don't. These are plants that are seeing things, and we have no idea what they are seeing, but they are definitely doing better. More so than plants only a half a mile away. What it is that's doing this, nobody knows. At least I don't know what they do in subsequent papers or there's stuff like that. But this is what we call fine scale local adaptation. It's not just local ecotypes, it's local ecotypes and steroids. These are Goldilocks plants, fussy plants. Half a mile makes a big difference, more than half a mile for these plants. What's going on? We don't know, but it says that sometimes you have to be real. So I just wanted to leave you with that as a tidbit because I was frankly quite surprised. One of the other things that we need to be thinking about when we do work with native plants and think about seed collecting and establishing new sites is that it's sometimes the case that the closest sources may be remnants on degraded sites where the population sizes are small, fragmented, that is, not close to other populations, or completely isolated, there's nobody nearby. This is a situation that often also co-occurs with genetic studies that show that they have less genetic variation. So remnant populations might be places where you want to go, oh, let's save the seed. But you have to be careful because of the fact that they're a remnant means that it's quite likely that they have suffered something called inbreeding depression. And this is a big deal in plant conservation. And it refers to a loss of fitness in seed of mates between close relatives. People who work in crop genetics and create inbred lines and then hybridize them to give you your hybrid garden varieties know about this. And they put up with this loss of fitness as they do the inbreeding because they're subsequently going to go back together and put different lines together. So it's less of a concern. But in nature, when all you have are pollinators and wind taking care of those issues for you, this can create a problem. You can see here is an example of what an inbred plant looks like. Plant from matings between relatives compared to the remnant output plants. So in inbreeding depression, as you get to have a very small genetic distance between parents, or sometimes a very small geographic distance between the parents, you have a decline in the fitness of the seeds, the plants that are produced from this. And that there's some kind of an intermediate situation with a somewhat greater distance between parents genetically and distance geographically that avoids this problem. What is that distance? That's not a subject that anybody wants to go into in any shared space. I don't want to talk about it at all. But inbreeding depression is a real problem when we're dealing with native populations that have been impacted by habitat loss, encroachment from urban development, agriculture, agricultural conversion of the native communities. So, whoops, jumping ahead here. One of the reasons why these happen, or why we get reduced genetic variation, is something called a bottleneck effect. What you heard of this? Bottleneck effect does not refer to how you feel the day after you had too many shots. It does not refer to the story you can tell the trooper after you've gone through a construction zone that's 10 miles long and all of a sudden we pegged you at 90 miles an hour, right? And those are different bottleneck effects. The bottleneck effect that I'm talking about here is something that is due to a sharp reduction in population size due to some random environmental event like an earthquake, a flood, or land use change. And as a consequence of this, only a fraction of the original genetic diversity that was present in the population is retained later on. Some people call this genetic drift. So here's the bottleneck example. The original population has all of these different colored beads, and we'll call them different flower colors just for the sake of ease and thinking about this, right? You go through this bottleneck event, this catastrophe that decimates most of the population, and there's only a few of them left in the surviving population. But as a consequence of this random event, there's much less genetic diversity there. There are no orange yellow guys left. Okay. And notice there's also a change in the relative amounts of the different genotypes. Previously, we had practically similar amounts of these ones. But after the bottleneck event, the surviving population is dominated by the blue flowered guys. Okay, so there's been a shift in the genetic composition of this population. And by chance, that can cause a big loss of genetic diversity. We have a homegrown example of this that is one of the studies that was done in my lab a number of years ago, but only published recently along with a bunch of other data. This is wild blue pine, Lupinus perennis, host plant for the primary blue butterfly, and near and dear to many of us in this group. And if you had the good fortune of working with this plant and ever collecting seed, you can see considerable amounts of just morphological color variation in the seeds itself. Sometimes you get white seeds, sometimes you get a lot of speckles, sometimes it's really, really dark. And one of my earliest students worked and studied 16 different populations across Ohio and Michigan. And she also measured the sizes of those populations. And you don't have to pay too much attention, just kind of like squint your eyes, and you probably see that on this side, where the populations are small, there are fewer color types of seed in the population. And as you slide up towards bigger populations in the many thousands, you have a lot more different types. Everybody see that? It's pretty easy. This is one of those graphs that doesn't let you do that. So small populations tend to lose genetic variation. And I can also tell you that now, in the case of this study, because it was done a while ago, that this particular part of that publication, some of those populations are gone, both because of human intervention, but maybe because of extinction due to intervention. So this is real, and it's in our backyards. On the other hand, local can sometimes benefit from non-local genotypes. In this case, movement of genes from an introduced seed source into a remnant population can be really beneficial. Can increase fitness of the new progeny that has both sets of genes running around in there. It can create what's known as hybrid advantage. So it's not just masking after inbreeding depression. Sometimes you just have the less vigorous, crummy genotypes that are left. And that's one of the problems with inbreeding depression in small populations. You can see from them. But if you bring in new genetic material, not only are you creating a more variable population, but the individual plants themselves now have genetic material from somebody else, right? I wasn't planning on saying this. Some of you met my husband is sitting here in the audience, who's 6'6". My whole family is like 5'8" and under. Okay, I have a nice tall son. Right? So believe me, being short is sometimes not cool. And by hybridization, I managed to create better, fitter, but these taller offspring. And we can argue about height as much as we want. But it's real, and it applies to plants too. So lost genetic variation is restored when you improve the quality of the offspring that are produced. There's a nice example of this. Some of you may have already recognized the plant that I was talking about here in this picture. This is Lakeside Daisy. Anybody in this room been to the Lakeside Daisy Preserve, the Marblehead area? It's a fabulous place. Lots and lots of plants there, right? It doesn't have small, tiny populations. From what I saw, the one time I saw it was at the very beginning of that preserve development. There wasn't a lot in it, but from what I've seen and heard, that they're doing pretty well. Turns out that this is a species that is self-incompatible. What that means is that if a bee visits multiple flowers on the same plant, none of those will result in mates, because it is not capable of crossing with itself, if you will, neither within a flower or among flowers within a plant. So this is like a genetic mechanism to prevent inbreeding depression from occurring. It has to find a mate that has a different genetic nature. Well, unfortunately, there are some places, in particular in Illinois, where populations of Lakeside Daisy have been so impacted that they never set seed. Only when they draw the genetic material from our Ohio plants will these Lakeside Daisy plants in the Illinois restorations capable of making seed. So this is sometimes referred to as a genetic rescue. So sometimes non-local, particularly if local has gone through inbreeding depression and loss of genetic variation, sometimes non-local, can be really helpful with NBD. So we said that inbreeding depression is bad. Well, guess what? So is outbreeding depression. Outbreeding depression refers to loss of fitness in the seed of matings with distant relatives. So sometimes two different is also bad. And in that case, we're talking about distant, non-local sources causing this reduction in fitness. And there's a whole lot of reasons why this occurs. And sometimes it's really hard to figure out which one is going on, and it takes a lot of work. And so people don't usually try to do it. One of the most relevant points that has to do with where we started in this conversation is that you have lower fitness of matings between individuals from differently adapted populations. So we started out saying that local is better because those nearby populations are adapted to that local environment. Well, one of the problems with outbreeding depression is that you have mated with someone and you have to have their genes and your genes, if you will, your meaning the local population's genes working together to function effectively, and they just don't play well together. They are adapted to different ecological conditions. That's what I meant by don't play well. Sometimes it's not just the interaction with the environment that is the basis for inbreeding depression, but it has to do with the fact that there are so many of the non-locally adapted individuals that are there that most of the matings are going to be of that kind. And you are creating what's considered to be a smaller effective population size, meaning the size of the population that is still well adapted is small. And that's kind of a hidden form of inbreeding that starts going on. So it's a little bit confusing. There's something that is called, and this is kind of out there, biology, if you will, breakdown of co-adaptive gene complexes. And by out there, what I mean is it requires thinking about biochemistry and the molecules that carry on chemical reactions. And the best way to explain this is that proteins in your body, for example, that carry on enzymatic reactions are usually the byproduct or usually formed from pieces of proteins that are produced by different genes that then have to get assembled in the right way to work together. So if one of the assembly pieces is made by a non-adaptive factory, if you will, and it doesn't work well, it doesn't fit well with the other one, then the entire molecule doesn't function. Does that make sense? Okay. So the non-local source and its fitness might influence, because of its genetic background, might influence the way the entire complex of molecules works together. And whoa, that's really bad. My bad. I hit the wrong button. It's pretty bad. There we go. We wanted this one. More than this one. There's another switch. One of the real problems is that you can't see this right away. It sometimes isn't apparent until later generations known as an F2. So if you started out, say, with a tall skinny plant and a shorter wide plant, and sure, they can cross and they make what look like a pretty happy pair of plants there. That's the F1, okay? But it isn't until those guys mate with each other and create the F2 that you start discovering that they're ruts, that they are not as fit, if you will, they are not as big. So outbreeding depression is very hard to detect, as you can imagine, from having to do multi-generational studies. Very few people bother to do look at that carefully and say, oh, yes, there's clear evidence about the expression. They just try it. But it is clearly operating in many of the cases where we are concerned about non-local genetic material. So that's one of the big reasons why we say local is better, because it can, non-local can lead to outbreeding depression in a whole variety of ways. So there's still some unknowns, many unknowns in the plant conservation community that professionally deals with issues involved in collecting seed and propagating native seed for carrying on restoration and for helping us create native landscapes. And one of these is, you know, it really depends upon the skill and knowledge level of the people doing this and what kind of training they're. And as evidence of this was a paper on there that I found. It's kind of interesting to me. They were working in Illinois prairie restorations. And in order to successfully restore these chunks of Illinois prairie, they went to these remnant populations. And different people collected the seed, and people who did this work took the seed from these different sources, from these different remnant populations, and then studied the genetic similarity of the resulting restorations to the remnants. And it turned out that the restorations that were created were genetically more, more correlated with who gave them the seed than with where the populations were located or the environmental factors. So some populations might have been more inbred, and that might have been the population where it came from. And the person did nothing wrong whatsoever. It happened to be an inbred population. But it certainly wasn't environmental factors. It had to do with where it was coming from and who had been collecting from those sites. And so maybe in another case, somebody only collected from 5 plants in that remnant population. And in that case, they caused a bottleneck effect during the course of collection from the remnant population. And so the resulting restoration ended up having the original. So these are difficult questions when you're trying to build up a stockpile of seed for people to grow native landscapes in their backyard and to restore the size of acres that people do. I mean, many people in this room are aware of Toledo Metro Park's Blue Nursery, which is a huge operation. And I would argue is one of the cutting edge top operations in this country because they do everything. They are just amazing. Those sorts of issues, I don't think are happening. But the take-home point is that you have to know what you're doing when you do this in order to build up these propagated collections of native seed sources. Keep in mind that similarly, depending upon where you go to purchase your seed or plants, they might have come from an ecologically mismatched site. You would assume that whoever is selling you this material is following these guidelines and not collecting from, you know, someplace out of state and marketing it as a local genome. Okay, so you need to be aware of these issues. Also, you need to realize that the conditions in a propagation garden or a new restoration are probably going to be different from the conditions when the original local native perennial seed established in the original population. Whether it's large or a tiny remnant, that might have been hundreds or maybe 10,000 years ago, depending upon what you know. And that's certainly not the same conditions in your garden or in your back 5 acres that you want to turn into prairie, or in the landscapes that we are trying to restore many. So there's a lot to think about when it comes to matching ecological conditions and how to provide the appropriate sources for restoration. And more so than anything else, people are now thinking about ecosystem resilience. So if we use seed from established remnants for new plantings, we want to make sure that we are thinking about the fact that they're going to have to survive increases in temperature. They're going to have to duke it out with invasive species that they probably maybe don't have an evolutionary history of interacting with, or extreme weather events. Okay, the cedar was really, really dry. A year or two before, in the month of June, rains nonstop. Okay, these are extreme weather events that characterizes climate change and what we're likely to encounter in the future that is unlike populations have had to deal with in the past. So restoration as a field and native plant landscaping started out with the idea of trying to bring back stuff that we had lost. And we're still trying to do that. But we also want to make sure that whatever we do back survives into the future, when that future is probably not going to be very much like the past at all. So these are huge challenges that people are looking at. So how should sources be chosen? There's kind of like three strategies that are, there's subdivisions of this that I won't go into, but three basic strategies that people have talked about. Local provenancing is what we've been talking about when we talk about the local genome. Provenancing is just a fancy name for where you come from, right? And that works for relatively undisturbed sites. So local populations with environments that are as similar to the site that you're working to restore as similar as possible is generally the best. So yes, local genotypes are probably the first go-to thing. But keep in mind that we've got to see how this works out. We've got to think about the future. Maybe we need to change what we're doing. And with that in mind, there are two different strategies. Some people have specifically developed what are called high-performing cultivars. These are native plant material that has been selected for tolerance to stressful sites. These are sometimes called native rocks. These are materials that people use for doing things like revegetating strip lines. Okay, really stressful sites are things that are really different from what the original, say, I'm focusing on prairie, but it could be any ecosystem, not just prairie. And most of our thinking about doing restoration is recreating something that's more natural. But when something is that stressful, it requires a slightly different approach. So maybe you just purchase some property that is former strip mine and you want to bring it back. You need to think about a different strategy for the seed source. And that might apply to something other than strip mine, depending upon, maybe you purchase something in an urban area that's a brownfield, former brownfield. And it's serviceable now because the lead levels are going up, but it might have all kinds of other stuff in it as well. So these are things to do. More relevant is the third approach, where people are starting to think about hybrid genotypes or mixtures of ecotypes. So maybe we need to draw upon local genotypes, plus bringing in a few herbs and spices, so to speak, that is, other ecotypes to help provide long-term persistence. Sometimes people have also thought about what climate change models predict for a future environmental condition for an area. So they fast forward the temperature and rainfall conditions expected in 2050. And on the basis of that, people might be selecting seed materials from an area to try to match the predicted future environment. So this is an area of hot debate in the community that works in this area, but it's referred to as predictive for climate adjusted provinces where they are adapted to predicted climate. And then using seed transfer zones, you can maybe achieve the same sort of thing without the fancy modeling. By sampling from within different seed transfer zones to sort of broaden across the region the environmental conditions to which your seeds might be adapted. And it's kind of like a way of placing your bets on what the future might be like. So these are all directions that enter into future choices. How should sources be chosen? Well, I'm going to give you some things to think about, but it really depends on whether you're backyard gardening or restoring or augmenting a relatively large area. And it also depends on whether you are really close to the natural area. If you're putting in a wetland restoration right next to, you know, a big naturally occurring wetland, like out of National Wildlife Refuge, or you're putting in, a prairie across the street from Oklahoma's Metro Park, you want to stick to local properties. Don't mess around. Because you don't want to be introducing non-local genes because a pollinator, a bumblebee from your yard, zips across the street and starts bringing non-local gene places. So you need to be a little bit careful, depending on where you are and what you're doing and what else is around you. But these are some general seed collection patterns that I sort of cobble together from a whole lot of places, and if it all kind of makes sense, it's kind of like diversifying your practices, but also being careful and conservative in other ways. So collect from multiple ecologically similar nearby populations and sets. Don't just take from one local genotype source. Get it from two different. Because you don't know whether the nearest one has suffered injury infection. So spread your risk a little bit. Collect seed from all ecological settings within the plant population. So you walk into the site, walk around a little bit, figure out where the shady spots are and where the sunny spots are. And if the plant that you are collecting occurs across that light gradient, collect seed from plants in all of those conditions. Because you're going to find that they are probably individuals with slightly different adaptations, and they will benefit you depending on how variable your site is, but also on what happens to your site in the future. Collect the large as well as small individuals. Everybody goes into a natural population, right? They see all these pretty flowering plants, and your eye catches the biggest one with all the big flowers and lots of seed forming, and it's like, I want that seed. Well, what if a little guy over here? Okay, so that seed might be valuable. We don't know why that particular individual is smaller. Maybe it's really good at dealing with drought, and it's smaller because it's in a dry pot. But its genetic materials might be valuable for disease resistance or something else. So think about collecting from across the variability that you see within the population. Don't just focus on the few plants, and don't just focus on the biggest. Here's a really important one. The 10 to 20%, and this is more conservative than what other people use sometimes. And this speaks to how much do you take from a single plant? What can a plant tolerate? So people have generally concluded that if you take 10 to 20% of the seed from an individual plant, that'll probably be okay. So take a little from many individuals. Don't just strip up one plant and then move on and strip another plant and say, okay, I'm done. Okay, distribute your collecting across many individuals. All of that being said, you need to think a little bit about your site. If your site is really disturbed, maybe the need for more genetic variation is even bigger than it would be if it was in really good condition and not very stressful. So once again, think about your site and the ecological conditions, not just how far away it is from the genetic source of the seed itself. And almost done. So I reproduced from the Wild Runs web pages some of the larger sources of seeds and plants with where you're likely to be able to find Northwestern Ohio local genotype material. There are others that are also listed on this web page, but I kind of tagged the ones that are most abundant. And somebody check me, did I get the dates on the upcoming plant sale for Wild Ones? May 20th and 21st in the spring. Sounds good. I don't know. Yeah, I think I checked my calendar because as soon as it was announced, I put it in. And there are other places that do this as well. But don't be afraid to ask if you are getting seed from someplace else. Ask, where did they get their seed from? If it's available. There's no reason why you can't ask that question. Know what you're buying. Particularly if there is no information about it. And when you plant, either plants that you buy or seed that you raise and then you outplant them, make sure you plant at least 3 to 5 or more individuals and plant them in such a way that they are plumped or grouped in order to provide opportunities for pollinators or the wind to produce crossing amongst those plants. Don't distribute them, don't sprinkle them amongst your yard. For looks sake, landscapers say planting groups of 3 to 5 or something, but from a fitness of the seed that might be produced by those plants point of view, you want to make sure that the bees are more likely to go to another plant of that same species than to move its pollen onto a completely different species because of the way you sprinkle really your garden plants. So think a little bit as you are gardening, as you are doing the native landscaping, about how what you do might influence the quality of seed in your garden space. And with that, we have to answer questions.
So when they took the seed from the Lakeside Daisy in Ohio, they don't know how to do that because they do not test each other to determine if it is not causing the problems. So you should do the best in the problems.
The question had to do with Lakeside Daisy when the genetic material from Ohio was brought over in order to restore fertility to the Illinois populations. I think they knew already that because the Illinois population was large and setting seed, that there had to be multiple mating types present within that population. I don't think at the time that early study was done that they had done a whole lot of genetic testing. I think they just assumed that because there were so many happy plants in Ohio that the mating types would be there. There was subsequent work done because it turned out that all of their restoration work didn't help a whole lot. There were site problems as well. And if I remember correctly, those restorations did not succeed for very long. So there could have been additional issues as well. But I think that encroachment and land use change was a bigger problem in those places than anything else. That just leads me to suggest that I think we have a similar problem here in the oak openings with cocoon, for example. So this is an issue that people should be aware of. Any other questions? I guess I could bring up the chat in case people out in Zoom land. Oh, I can't bring up the chat, can I? Oh, wait, yes, I think I can bring up the chat. Okay, now I can see the chat if we haven't lost people from out in Zoom land who wanted to ask questions.
Yes. How your result was finding the area and real green, but well intended. But I was wondering how you match your site to your seed. How you kind of match them.
So
Without scientists,
it's don't know this 100%, but Ohio Extension would probably facilitate the process of getting your soil tested. Am I correct?
yes, there is
Yeah, OSU, Ohio, I mean, so OSU is still Michigan, right? Or Michigan, those places will help you figure out, how fertile your soil is, what its pH might be, kind of soil it is, which will affect, how much it will hold moisture and things like that. Those are kind of the standard places that everybody can start and ask about, so. And so when you consider seed sources, you know, and first of all, you might want to use this to help figure out what species you want to plant, right? Because if you have clay soil, you're not going to put, for it'll look great for 2 or 3 years and die. It just doesn't do well in any place. And find other places that have seed sources that might be similar environment. Something that I didn't talk about is work that you did on these lupine populations in Ohio and Michigan. And if I remember, some sites have slightly higher pH, and those populations also had more seed production per plant. And if you know, if you need to change the pH of your soil, maybe more appropriate for what you're growing, or if you are in a spot that has a slightly higher pH, maybe some lupine from over the border would do really well in that situation. But if you're in a place where the pH is really, really low, then you probably want more Ohio type. That help? No one in Zoom land wants to ask a question. Thank you all for your time.
I can also set a proper welcome to everybody. I was so kind of distracted when we got started from all the technology stuff. So if I didn't say that, welcome to everybody, those who are members and guests. We appreciate all of you coming out to Orlando for our first in-person meeting since February 2020. So it's pretty exciting. We do have some work prizes that Andrew has organized, so we can probably end the Zoom meeting on that. So Helen, if you want to say a farewell to the Zoom, please.
Oh, you know what? I think I shut my microphone off already. Oops.